Integrated Sciences Academy, and I'm not talking about integrated sciences, so I'll mention that for a minute, and then I'll talk about color science, which is what I spend most of my time doing. Integrated science is where we keep our multidisciplinary programs, and color science, as you might imagine, is one of those. I'll explain what color science is in a moment. But the other one I want to mention, since I have a room full of high school teachers, is we have a one-year program, a first-year program called Science Exploration. And that's for students that don't want to choose their major when they're a senior in high school, when they apply to RIT, but would rather say, I think I want to do science, and can I go to RIT and spend a year exploring science and deciding what I want to do? Which I'll get on my little soapbox, that's what pretty much every other university in the country does. But we like to get students into their majors as quickly as possible. So the Science Exploration is a route to do that and not get behind. So we advise them strongly. If they know they're more into chemistry or biology, we send them that way a little bit and so on. But it's my favorite program because it's in my school. And I like all the programs.
Are you going to explain more about that? I have a question.
Ask a question now because I don't have more in my talk.
Okay. What would a typical Explorer Science major take in their first semester?
I don't know if there is a typical one, but basically we line them up with a physics sequence, a chemistry sequence, a biology sequence, and a math sequence. They don't necessarily do all four. So that's where we let them say, well, you know what, I'm not going to be a biologist. So then they might do physics, chemistry, and math, and the liberal arts. So there's advising to pick them. But basically, we try to get them the intro sequence in each of the four general areas. And that's what many of our programs are doing anyway. Not all of them, but maybe 2 out of 4. They all have math. So we try to line them up with where they're leaning. But if they really don't know they could do all 4 sequences, it's possible. And they're busy. I like to say they should be the very best students in the college because it's not an easy program. A lot of times it's thought of as the default for the kids that can't make up their minds. It's really for the kids that know they want to explore and figure things out. So it can be very challenging. The other thing they all do is a project in the first year. It's basically a research project as a group, very hands-on. They get to define the project with the instructor. They've done things like explored for life on Mars, where they get meteorites from Mars and then they analyze them and see if there's any things in them that could have been signs of life. And they do that for the whole year. And they've given presentations at conferences and at Imaginary Key and things on what they do. So they get hands-on multidisciplinary project freshman year as well, which helps them explore. Let us know if you want to know more information. You can just contact this program and they'll get to me or whatever. All right. I'm going to make the lights a little bit lower if you don't mind, because I have a few. Let's see if I can without putting us in total darkness.
That's fine.
Is that okay? If you start going to sleep, let me know. I'll try not to let that happen. I have a few visual things to show. I'll give my first disclaimer, I'm not a mathematician. And my second one is I'm not a math teacher either. I'm a scientist and I teach only graduate students for the most part. So our program in color science is a PhD program and a master's program. So it's pretty rare that I am in a classroom with undergrads other than a visiting lecture now and then. We're starting some neuroscience courses in our program that I'll be involved with the undergrads. That'll be fun. We don't have a bachelor's program yet, but we're hoping to do that. So I'm a very visual guy. And normally in my talks that I give at conferences, and even for a large part in my lectures, I leave all the equations out and show visually what happens with what we're doing, and people can read my papers if they want to see the equations. For this, I didn't want to do that because I wanted to give some examples of the different math that we use. So I grabbed a bunch of things from a bunch of different places. I have way too many slides, so forgive me in advance. I'm going to skip over some. I'm going to see how it goes. Some are just to flash up some images to show you. And I just want to give you some examples of how we use different sorts of math and statistics in color science. So first, preview of the tour if you're coming on the tour. One Cell Color Science Laboratory is the name of our laboratory. It's had that name since 1982, which happens to be the year I came to RIT, but I was a student then, not on the faculty. Long story how that all happened. It's named after this guy, Albert Munsell, and you can tell he's old. He was old. He died in 1917. He developed a system. He was an art teacher, and he developed a system. This is just a picture I took in the lab of some of the atlas there and the more modern Munsell book of color that you can still buy. And he basically made a dictionary of color. Because as an art teacher, he was very frustrated that he couldn't communicate color precisely to his students or other faculty or anybody. So he basically made a dictionary and said, if I have a certain color, this one, I can look at it and see what it is, and I can give it a certain designation. It has 3 numbers, a hue, a value, and a chroma, or the names that he used. And that became the Munsell Book of Color. He had a company. Company made all sorts of products like crayons. There's Munsell crayons in the past. That got bought out by Binney and Smith, who makes Crayola crayons. Turns out they bought out every crayon company in the United States because there were no antitrust laws back then. That's why we all have Crayolas, up until a certain point anyway. I always thought it was the other way around and that Munsell should have kept the crayons, but they wouldn't have been a big crayon company. The company was eventually sold. There was a foundation that the family controlled, and they existed to support students studying color science, the science of color. In 1982, the foundation voted to dissolve itself and transferred all the assets to RIT. I'd like to say it was billions of dollars. It wasn't, but it was enough money to put the name on our lab and assure that we have a lab where people study color science forever. So I can misbehave, they can fire me. Even though I have tenure, they can still fire me if I misbehave. But there will still be a color science lab and a color science program here because the program and the lab are endowed, which is what that bought us and why we like to point out Munsell. So there's our founding sign. You'll see that over in the lab on the hallway. Objectives, education, applied and fundamental research, measurements of color. In the 80s, when we started, measuring color, things we do with our phones now, wherever I set my phone, or very inexpensive instruments to calibrate our displays and our cameras. Big companies didn't have the instruments back then. They cost hundreds of thousands of dollars, so companies would come to us to do measurements for them. And that's what my first job as a graduate student here, is I did measurements. Our stipend was a whopping $2,500 a year. In the 80s, that still wasn't much, but the director of the lab then said, okay, you do the measurements, Mark. We know that's boring. I'll give you half the income. So every measurement was like $250. I got $125. And that's how I survived for a long time. I love that guy. You'll see the room we named after him. We don't do that as much, but it used to be a big service for the lab. We still do it for our research. And then working with people, industry, academia, a lot of interactions. We do research, a lot of research with industry, and so on. Keep going. Demo. This is what I do. All of my research in color science, I study visual perception, specifically. People in color science study everything from the production of light to materials and how they interact with light and making them the right colors, to the human visual system and how we perceive that light and interpret it as color. Color only exists up here. No matter what a physicist tries to tell you, there is no color out there. These objects aren't colored till we look at them, until there's light to bounce off them, and then us to interpret them. Whoops, I'm getting ahead of myself by accident. Bowl of fruit, nothing magical here. I'm gonna virtually take a cyan filter. So imagine I actually have a piece of gelatin that I could hold up in front of the lenses. If I just projected white light and held it up in front of the lenses, you get this kind of cyan colored light, bluish green light. So imagine that filter. And imagine I'm going to take a pair of scissors and cut it out in the shape of a banana. So this is the quiz. And I expect my color students to always get it right, but I don't expect you necessarily to. I hope you do. If I take a cyan filter and put it over the yellow banana, what color is the banana going to become? Green?
Is it opaque or translucent?
It's not opaque, it's transparent. The light's going through. Green's the right answer. The cyan filter basically is taking away red light. It's good old subtractive color mixing. The yellow image has already taken away the blue light. This actually is math. Minus the red light from the filter, minus the blue light from the banana. Neither of them take away the green lights. When I put the filter in front of the banana, the only light that isn't taken away is green. Simple physics really. And here it is. So cyan filter in front of the banana, green banana. And all the students are like, yeah, of course. Physics works, math works. It really, we write these equations down. We do it mathematically. What? Sad looking banana?
As a banana ripens, the filter is removed.
Yes. Yes, I'll go back. They're nice. Yeah. That's not how bananas ripen, but now I'm going to do the vision science part of it, the perception. Instead of putting the cyan filter just over the banana, I'm going to put it over the whole image, front of the lens basically. So physics is the same. Yellow banana takes away blue light, cyan filter takes away red light. That green banana is still going to be a green banana, right? So there's the green banana. Now I'm going to put the filter over the whole thing. What color is the banana? Why did it change color? I didn't cheat. Physically, it's the same. I'll go back and forth. Stare at the banana. It's not changing. The rest of the image is. It's a perception that's changing. Because when I make the whole image cyan, your visual system adjusts to that, just like it's doing all the time. When we turn these lights down, you become more sensitive to light so you can still see me. When you go outside, it's daylight, it's bluish. You become less sensitive to the blue light, more sensitive to the red light. You turn on a tungsten light bulb at your home, it's very reddish. The opposite happens. I made the scene bluish. You, your eye, and your brain adjusted to that very quickly. Relative to that new white, which is bluish, the banana is still yellowish. That's all perception. Physics is identical. So if we just did physics, my physics friends would say the banana didn't change color. Perception people say the banana changed color, because it did, because color is a perception. I'll go all the way back just to show you one more time. Original scene, kind of normal white. Put the filter over just the banana, turns the banana green. Put the filter over everything. Shifts that image towards cyan. You adjust for it. Banana's still yellow. So we're trying to figure out how this all happens and literally write equations to model that. Yeah?
So that example, does that have anything to do with like the dress and
It
all of
does.
that?
Yeah.
Okay, cool.
I'm not going to explain the dress
No,
today,
I
but
I'm just curious too, so-
basically the same explanation. When the dress came out, my older daughter was a freshman at college, and she texted me that morning and said, "Dad, explain this." And she just sent a picture, and she's like, "All my friends, we looked at it, we all saw different colors." I'm like, "Well, you all looked at different phones, they're not calibrated," which is my standard answer, right? The displays suck. And she's like, "No, Dad, we're not that dumb. We all looked at the same display and we see different colors." So then I'm like, "Oh, I gotta look at this." And then I got embroiled in a whole bunch of interviews and things that day, of course. But yeah, it's basically the same idea. It's how your visual system adjusts and what assumptions you make about that image. Different people make different assumptions about what the lighting is, so they see different things. Yep. Okay, so what I'm gonna run through. A physical model, how we mix colors. The integral, which is our main integral of color imagery. The peeps, which is the people, how we model them. Displays, scaling, space, not outer space, but how we make a space out of color like Munsell did in that book. And then the big model that we've been working on. I don't know if I'm gonna have time to do all these, but I'll give it my best shot. So physics. We have a whole graduate class called color modeling. And what we mean by modeling then is not, building chips, it's mathematical models. How do we take the stuff we make, displays, plastics, paints, whatever, and predict what color we're gonna get when we vary whatever we can control, whether it's amount of pigments, whether it's amount of dye, volume in a liquid, whether it's digital values on my phone display or computer. We have things we can control. When you go to the paint store, I just did this over the weekend, get some paint mixed. The people that mix the paint know a thing called shots. They say, "Oh, you want that color? It's 3 shots of brown and 2 shots of yellow." That's all they know. The shots are very controlled, basically drops of paint that go in a white base. Well, somebody like me wants to know, what perception are you gonna get? This is just like kind of show our color space. I wanna make a certain color. I wanna match the pink lid on this cup. I can measure that and tell you what color it is, but I don't automatically know how much paint to mix to match it, or how much light to mix on my display to match it. That's the modeling part. Measuring it, then inverting the math to say, how do I make it? So we need laws, mixing law, like a physics law. Ideally one that gives us a linear relationship between something we can control and the color, something we can measure that correlates with color. Scalability and additivity, that means if it's linear, well, I don't have to explain to you what it means, but if it's linear, it scales. You double whatever you're controlling, you get twice as much of the effect. Additivity means if I mix red light and green light, adding some red light doesn't affect how much green light is there. If I mix two pigments into my paint, putting one in doesn't chemically react with the other one and take it away or something like that. They're independent. This is a display. So the top one, this is wavelength. Think of the rainbow. Blue end, red end, all the hues spread apart. This is a blue phosphor on a display, the RGB, at various intensities, various digital values being sent out to the display. And you can see the curve increases. You get more light when you send a higher value out. That's how it works. But if I normalize those, they all fall right on top of each other. So the display is scalable in light intensity. It's what we call an additive display. The light adds up. Turns out it's nonlinear with the digital values for obtuse stories about how old televisions used to work. The physics of the cathode ray tubes was nonlinear. So these things are built into modern display, curves like this, to mimic old-fashioned TVs, because all of the content works that way. But if we characterize that nonlinearity, characterize this scalability, we can then predict how to make a color on the display, which is done all the time. Pretty straightforward one. If we're mixing dyes, it's not linear in light. And the simple way to look at it is, if, I'll look at this one. If I put 1 unit of dye into, say this is a little cuvette that I can put dye into, I get a certain amount of light through. If I put 2 units in, so let's say I get half the light through 1 unit, 2 units, I get half the light, then I get half the light again, so I get 25% of the light. So it's not linear anymore, it's a geometric scale. And we model that, basically you have to take a log. So you get log of the 1 over the transmittance is a thing called absorbance. Those things add up. So simple mixing of subtractive things like the filter I started with, almost as easy as light. Not too many real materials work that way. This just shows that if you take that log, so transmittance is kind of what we see, the percent of light we see. Absorbance is in the log of 1 over transmittance. Those scale when you normalize them all the same shape. That's where we have our scalability. We can add up the pigments. We can mix colorants just like we mix lights. Real stuff does more complicated things. And paint, for example, when you go to the paint store, that's not a simple add up the light or add up the concentration of dye in a clear material, because the light is both absorbing light, kind of the middle one, transparent film on an opaque support, that's like clear ink on paper, just absorbs light like a filter, but it's also scattering it. That's what makes it opaque. Opaque means you can't see through it, you can't read through it. So you have to model both the absorption of light and the scattering of light. Long story short, there's the Beer's law model, which is the simple absorption one up here. That's Beer's law. It applies to beer, by the way, as long as you get the bubbles out and no condensation on the glass. Experiments have been done. Kubelka-Monk theory applies to this. And what we have, instead of just an absorption coefficient, we have an absorption of coefficient and a scattering coefficient. They happen to follow this equation with respect to reflectance, and they add in these values. So you have to solve these equations for the pigments for K and S, and then you can add them together like this, where I want to know the mixed up paint that I just bought at Home Depot. They have to know the absorption and scattering of each pigment that goes in the paint and how much they will need to do in order to get the desired result. And you can see that's not going to be linear. And then there's another step to relate that to the color that we get. That's about as complicated as it gets for color mixing, but we still haven't talked about the perception. And as you might imagine, there's sort of ad hoc computational models people use to it. They say, you know, the theory doesn't work perfectly. I'm just going to measure everything. And I'll mix all the possible paints together and measure them and make a giant lookup table. It's really cheating. It's when you don't understand the physics of what you're doing or the chemistry. And that's done a lot too. A lot of devices are calibrated that way. So let's say we get that. We get physically the light. That's what those models are about. They're telling us what light reaches our eyes. We then have to figure out how we respond. So that's where our little bit of calculus comes in, our integral. So it's based on two concepts, one called trichromacy. Have you ever heard somebody say that your visual system's three-dimensional with respect to color? Or that you have three types of cones in your eye, cone photoreceptors? There's three types. Cones are the ones that respond to high levels of light. That's where we see color. Roughly sensitive to red, green, and blue light. There's three kinds in normal people with normal color vision. I always make mistake and say with normal humans, which is not what I mean at all. People with normal color vision. People who have color deficiencies or color blindness might have 2 or 1. And there's some rare people that have 4. Won't get into that, it's a genetic thing. They don't see 4-dimensional color though, they still only see 3-dimensional color because the wiring in the brain isn't prepared to handle 4. Maybe someday. So trichromacy is the threeness. There's 3 variables we need to control. That's why our displays have red, green, and blue light. That's why I could talk about the banana as subtracting red, subtracting blue, leaving green, and I could kind of tell you what color you have. Threeness is important. We only need 3 integrals. Metamerism is the second point that's important. And that's dealing with the fact that your receptors in your eye can't tell me what wavelength the light is. Right? You've probably heard it one time or another. I just showed you. I said, imagine the rainbow from blue to red. The different wavelengths tell us where we are on that rainbow. We can't look at something and figure out what wavelength we're looking at because our visual system doesn't sense that way. It senses broadly red, green, and blue and gives us one answer for each of those. That's called univariance. We're not spectrophotometers. We're not analytical instruments that figure out the physics of the light. We just get these 3 integrated signals. That's where the integral comes from. Those 2 concepts let us do what we call colorimetry. The basic idea of colorimetry, color measurement, is that you have one color on one side of a split field, imagine. On the other side, you have some primaries you can mix together. They don't have to be something technically called a primary. They just have to be 3 independent lights so that I can adjust the amount of those lights to match the color I'm interested in. By adjusting 3 lights, I can match any color because I only have 3 dimensions of variability in my retina. So if I only have 3 variables in my eye, I only need 3 variables in the world to make all the colors, plus or minus a few that might be out of the range of a system. That's why color television works with 3 primaries. We make that match with standard primaries. We measure how much of each of them. We've done colorimetry. We've measured the color because I can go back and match it again later. I could say I want to make my pink cover in paint. I visually match it with my red, green primaries, red, green, blue. I go to Home Depot. I say, I want you to dial in this amount and make me a pink paint that matches it. And they can do it. Now, you need me to do that because I made the match. The mathematics of color science removes the me from the equation and makes it mathematics purely. But that's what's going on when you ask for a certain color. So those 3 values. There's this guy called Grassman. He did a little algebra for us. He looked at these additive mixtures of light and said, you know what, if A matches B and C matches D, and if A matches B and C matches D, we use the word matches rather than equals because physically they don't have to be equal to match. There's more than one way to make a color. That's the metamerism point. So we say matches, or sometimes we use an equivalent sign. So the equal sign with 3 lines. But if A matches B and C matches D, A plus C match B plus D. You take 1 from each side, mix them together, and the matches will hold. Lights will add up, even though they're different lights. Subtractivity, if A plus C matches B plus D, is kind of the inverse here. And C happens to match D, then A matches B. We know this. And transitive, if A matches B and B matches C, then A matches C. It's the same properties you have with numbers. It's basically telling us algebra works in color matches. And proportionality, if A matches B, then K units of A will match K units of B. The visual system is linear with respect to absorbing light. It's completely nonlinear after that, but the absorption is linear. That tells us we can do algebra. That tells us we can figure out how much light we have at each wavelength and add it up like an integral does. We do these matches. Color C is matched by R units, capital R units of the red primary, G units of the green primary, and B units of the B primary. Those amounts are called tristimulus values. That's colorimetry. If we do it all the way through the spectrum, wavelength by wavelength, through the rainbow, we can do those matches. And I already said this, but basically wavelength by wavelength, if we know how much light it takes to make a match at each of these, and we know what Grassman told us that we can add them together, then if we want to know for the whole light all the way through the spectrum mixed together, it's an integral, weighted by the visual response. So the additive mixture turns into an integral. These are the human visual functions on average, telling us the response to blue light, green light, and red light to match a color. 1 unit of energy at each wavelength. So I said this is the blue end of the spectrum, takes more blue than green or red. This is the red end of the spectrum, takes more red than green or blue, and so on. Green in the middle. At the ends we can't see. It's infrared or ultraviolet. Can't see those. They're actually not light. Little pet peeve, there's no such thing as ultraviolet light. There's ultraviolet radiation. Light is something we can perceive. That's the range of light. We integrate across those wavelengths. We know how the visual system responded to the light. Therefore, I know how much I would have dialed in to match my paint cup. And I can go to the paint store and said, I would have dialed in these 3 integrals. So make paint that will produce the same integrated values. They use their Kubelka-Monk theory in the computer. The paint has a certain reflectance. They assume a certain light source. They don't know what light I have at home, but they assume one. And they make the integral match whatever I asked them to match, whether it's a paint chip from the company or whether it's something I brought in. So we can do linear transforms on these. So these are spectral sensitivity functions of the visual system to a certain set of primaries. I apply a 3x3 linear transform to them, I can make another set. XYZ happens to be a standard from the old days, 1931, where people didn't like negative values. So they derived a linear transform where none of these went negative. It's a long story to explain why they can be negative. Trust me, they can. Why do you think they wanted to get rid of negative values in 1931? People have to calculate these integrals over and over again. So why did they not like negatives? We don't care at all anymore, right? Because they're in a data file, program calculates them.
1 over e to the 1 over x.
No, didn't matter there. It could. That's a good insight. I don't usually have math teachers. Literally, these calculations were done by rooms full of people. The old guys I knew would say the rooms full of women because they happened to all be women back then.
Yeah.
On adding machines, those kind of adding machines. If the people missed 1 or 2 negative signs, you got completely the wrong color, and they had to hit it each time. So they literally, in the international standard, got rid of negatives. They didn't make it less accurate. They derived the proper way to do that. They got rid of negatives. They did a few other things. So those are standard functions that we still use today, almost 100 years later. We've made some better ones. So I don't have to explain linear transforms to this group. This is what I would do for students trying to understand why changing from one set of lights to another is a linear transform, because the match to any set of primaries is an additive amount of the 3 amounts of the other 3 primaries. This is just showing 2. But if I know the amounts, then I know the matrix of C's there. I can go from one to another or back all day long. This shows it. RGB, which is the plot on the left, XYZ on the right. There is a matrix to go from one to the other. Yes?
Quick question. This goes with like websites and RGB
Yep.
codes for colors.
Yep.
I've heard this, but I don't know if it's true. That there are certain colors that you can't create just doing an RGB code. I'm wondering if that's true or why certain colors get left out when you try to use these 3 numbers.
Well, historically, there was a limit to the number of combinations you could use. So there's these old things called web-safe colors.
Okay.
That's not relevant anymore. You'll
Okay.
still see it. But there's another thing. That pink top may not be able to be produced on my display. So if I calculated the RGB to make this, it might say, you know, you really need negative amount of green. There's so little green in this, I can't make it. And then you get, instead of values from 0 to 255, you might get one that's greater than 255 or less than 0.
So it
And
just
that
has to
can't be
like a
produced.
physical limitation.
That's a
of the
physical
white light.
limitation, right?
Okay.
It's none of this math. The math keeps working. The negative value is the right one, you just can't make it. You can't make negative light on that display. You might be able to get another display, like a cinema display, digital cinema display that could produce it. So there's those two things, but the web safe codes is not relevant anymore. You can make any of the colors on that are in the physical gamut of the display.
Thank you.
This just shows the idea of transforming those primaries. Going from one set of RGB to another, it's a rotation of the axes. That's all it is. And then here's a diagram of the integral. So what we do, light source, this is an average daylight. Object, this is one that's reflecting yellow, green, red light, absorbing blue. Multiply those together, that's how the physics of the light works. You get the visual stimulus, reaches the eye. Have those three response functions, multiply by that, then integrate them and get three numbers. And those are the three numbers that are the basic metric of colorimetry. This is just showing it again another way. Light source, object, visual response, three numbers. That lets us do color matching. We do it as summations rather than integrals because we only sample the spectrum. We never have it continuously. Same equation. One more flow chart, same thing. Light source, reflectance, stimulus, visual response, integrals. Now you notice I haven't said one thing. You might not notice because I was going fast. This tells us how we match a color. So I could figure out, they're called XYZ, the XYZ values to match this pink color, but with those three numbers, I don't know it's pink. I know what matches. Now think of the banana. There's a reason I showed you the banana. Besides that it's really cool, the banana with the filter on it, whether the filter was over just the banana or the whole image, exactly the same XYZ values. To physically match that, I would dial in the same numbers. If I had time, I could show you on my display that it's physically showing you the same RGB values. Your brain saw something different. This tells us about matching. So if I want to make that green banana, I can make it as long as I view it in the same environment, either in the slide covered with cyan or not, and it'll look okay. So there's a lot more to colorimetry. I'm going to talk about some of it, trying to go from matching colors to the appearance of color, what it actually looks like in an environment. So a little bit more about people. This is my research. Got 25 minutes, I'm doing okay. So we're all different. Everything I just told you is true on average. Those visual response functions are averages that have been standardized internationally. Everybody uses them, they're great. How many of you think you are the perfect average observer? None of you. I've measured lots of people. Some are pretty close, but the example I like to use, the average American family now has, I believe it's 1.9 children. It used to be 2.5. 1.9. How many of you have 1.9 children? Yeah, I have two girls. Sometimes I think I have 1.9. Sometimes I think I have 3. But, you know, they're quantized, right? Nobody is the average family. Nobody is the average observer perfectly. So people are individual. We know a lot about genetics now. These are showing two kinds of cones, the long wavelength ones, which we call red colloquially, and the middle ones that we call green. It's not showing the blue. Some little transpositions on a couple of genes, and look at how the wavelengths shift. The wavelength of sensitivity, depending on what gene you got from your mom and dad. Yeah, mom and dad in most cases. So there's these shifts, and I dreamed when I learned about this 20, 30 years ago. I thought, wow, so someday I'm going to be able to like take a cheek swab, run over to my friends in biology, sequence their genes, and know their visual sensitivities. That would be so amazing. And 30 years later, I still think it would be amazing. Problem is, this doesn't tell you the whole story. There's a lot of other stuff going on in your visual system. So there's nature and nurture. The nurture messes up your visual system. So this doesn't directly tell me what color you see. but it's a hint that we're all different, and it's one of the reasons we're different. We have to do some other modeling, not just the genetics, to get it out. So this is a model of an individual observer. So we have the 3 cone responses. Think of those just like XYZ. They depend on the alphas, the light absorbed by the cones. Those are the things I just showed you. They depend on this thing called the macula. You have a little protective yellow filter on the most sensitive part of your visual system. When I say look at my hand, you move your head and your eyes to line up where you have your best visual acuity. So if you look at my hand and try to read the rating, even on that slide over there, certainly on the wall, you can't do it without moving your eyes, because only a small region has good acuity. The macula is a protective filter over that small region. In case you happen to glance at the sun, it absorbs away the blue light that can damage your eyes quickly. That's different in all of us. You can make it different by eating more carrots, because keratin is part of the yellow pigment in there. So you eat a lot of carrots, first of all, your skin starts turning orange, and that's when you need to worry about your color vision after your skin's turned orange. It's not that big of an effect, but it's real. But we're all different. Then we have the lens, ocular media density, function of age. As we age, you've probably heard of cataracts, right? Yeah. Cataracts are when your lens becomes opaque and you can't see through it anymore. You get cataract surgery, they pull out your old lens, the one you accommodate with and can focus near and far, put in basically a plastic one, not really plastic, and then it's clear and you think, wow, I can see again. Then you have to put on the glasses in order to focus. They're fixing that, so you don't need the glasses anymore. But that is the extreme aging. Your lens turns opaque. As we age, it's getting yellower and yellower. So at my age, my lens doesn't accommodate very well anymore, so I wear bifocals so I can read close up. When you're young, you can put the newspaper, if you had a newspaper, you can put your phone right here and read it, because your lens is so flexible, you can accommodate that close. As you age, the near point moves away. All that time, it's getting yellower and yellower. It's like I put on a pair of yellow sunglasses. Why don't we notice? Green banana. We adjusted that cyan cast of the image very quickly. This yellow is happening over our entire lives. We're adjusting to it all the time. So we don't notice it until we get a cataract and they pull out a lens. We're like, wow, look at all that blue I was missing. Those 3 things tell us the visual sensitivity. So we have a model, the 10 to the minus D. We're taking the density of the lens and macula, turning it into transmittance, like the lens filter, multiplying by the sensitivities, and we get there's a 4th one there for the rods, but we get the 3 responses of the visual system. I'm going to run through some of this quickly. This is what the macula looks like as a function of wavelength. Lens density is absorbing more light at the short wavelengths. And we play a Monte Carlo game. So we do a Monte Carlo simulation. We have pretty good statistics on the cones in the population, on the lenses in the population and how they change with age, and on the macula in the population. So if I want to build an observer, rather than taking someone in the lab and torturing them for a couple of weeks to measure their sensitivities, I can just roll the dice and say, okay, I'm going to randomly choose a set of cones. Come back to that. I'm going to randomly choose a macula and randomly choose a lens and use that kind of model to build the visual responses. From that, I can build up populations, compare them to real populations, and basically simulate the world rather than measuring them. The next point is it doesn't have to be a normal distribution. We use different distributions to sample these variables. I already said this, randomly select those, build the what we call color matching functions or CMFs. But how? We'd like to know the appropriate random distributions. In some cases we do, in some cases we guess. There's a great model of the density of the lens as we age. One factor in its transmittance is age dependent, one is not. It also varies with exposure to UV and things like that, but we can predict the population. We actually took the US Census data to have a population of ages in the US, plug that into the lens model to get lenses for a given age, which are the average lens for that age. To make individual variability, we randomize the age by plus or minus 5 or 10 years, something like that. And this shows that density. So density means it's absorbing light. At 1, it's absorbing almost all the light. At 0, it's not absorbing any. It's absorbing blue light. And those are curves for ages 20, 40, and 60 years old. You get an idea how it changes. Little tiny 1% changes in these are perceptible. These are big changes. The macula, similar model. We scale it with a normal distribution. This shows the absorption of the macula, similar plot. This is that protective yellow filter. The mean plus or minus 3 sigma. Again, huge variation. Some people it varies from left to right eye. So if you ever noticed colors look different from the left to the right, like if you're a painter or something, it's real, it can happen, it's no big deal. Might be frustrating at times. Cones, I talked about the genes, they don't work. I'm gonna flip through this. The phenotypes, us, show continuous variation. We did some stats on that. Gave random offsets, get these kind of functions similar to what I showed with the genes. And these are all the components together, put the model together. Here's 1,000 observers. And it's kind of a bad plot because once you plot 1,000 lines on top of each other, it looks like it's just a big wide line. There's really a lot more near the center than there are near the boundaries, kind of normally distributed. But remember, I said about a 1% change in these curves is noticeable. There's big differences between people. So when you go shopping with your spouse or significant other, and you say, wow, those curtains match our paint perfectly, and the other says, they're not even close, it could purely be a difference in the physiology or color vision, and nothing else. It happens all the time. We are different.
I'm gonna use that.
You should use that. If nothing else you learned this week, it'll save you an argument. Just remember, it looks good to me, and that saves the argument. My wife has a master's degree in color science, so I can't get away with any of that crap. But so this is some old data that shows that kind of variation as well. This is real. We simulated this, but it matches the data. If we do a little trick here, these are what we call metameric curves. There are 2 different reflectance curves that give you the same color appearance under a certain condition. That's part of this problem. That curtain might match the paint under daylight and then not match under incandescent light because they're not exactly the same material, as well as not match for you and your wife, for example. So we made a bunch of these, and each of these, this is worth explaining because this is the visual impact of all that I just said. Each of these are 2 different physical stimuli, top and bottom. These 2, just look at this red one, red here, for a moment. For a standard average observer, they all match. The 2 on the left always match. The 2 on the right are 2 different observers from our population of 1,000 taken to be at the 95th percentile, extremes. So the standard observer says, yeah, paint, drapes, they match. I say orange, you say pink or whatever, because we're 2 different people if we happen to be at the extremes of difference. What's cool about this, especially this one up here, those are completely different color names. Most variability we see stays within a name. It's green for me, it's green for you, but it might be a bluish green for me and a yellowish green for you or something like that. The names don't generally change. That's why we have so few color names, as it turns out. But I just want to show you people, these are normal people. These aren't colorblind people. It goes crazy for colorblind people. These are normal color vision. You can make colors that will shift quite dramatically. That's the lesson. And we do that, of course, all mathematically. I just said all that. This is a scatter plot of people making those kind of matches between a print and a display. And the scatter we see for 20 people, don't worry about the units. This is the space that I'll talk about for just a couple minutes. And these are the data points similar to that for our 1,000 simulated people. The thicker curve is that 95% confidence interval put on the simulated data 30 years later, 20 years later. And the lighter curve is a 95% confidence region on the actual data. Now you might say, well, they don't match up that well. Physicists might say that. Somebody studies perception goes, oh my, that's awesome. They almost are the same orientation. They're the same size. We didn't do any scaling. So that means our Monte Carlo model is a pretty good representation of that population. In fact, if I were to bet on one, I'd bet on the model because that was only 20 people. I had 1,000 people to do the simulation. So we can do it. We can predict the variability. That's the news. Okay, displays. We've already talked about this. Displays are additive, so you add up the amounts of X, Y, and Z. They're nonlinear, so we have this thing called gamma correction, fix that nonlinearity, and we can predict the color from the amount of RGB in the display. I won't revisit that because I have more fun things to do. You might have heard of sRGB. It's standard RGB. Standardized by people from HP and Microsoft in the 1990s. 2 out of 4 of them came from our program. I don't know if I want to take credit for that or shun them, but it is what it is. If you have a digital camera, just a regular, your phone camera, whatever, chances are probably 99% that the images are stored in RGB values called sRGB. The assumption was, we don't know what display you're going to look at them on, but these represent the average computer display in 1996. Therefore, it built up and it became a standard so that the internet colors would look reasonable on different displays. Now, these manufacturers are so good at building displays and calibrating them, it doesn't matter what your display is. They get an sRGB image, they say, okay, it's sRGB. I'm going to do all that math that Mark just talked about and adjust it to look right on my display no matter what it is. You know, our phones go way beyond these, but it's a standard that lets us communicate. So it's essentially a system of color measurement. And this is the numbers. We don't care about numbers today. So scaling. I do want to stress this. Scaling is where we get into what things look like rather than just that they match. So we do a lot of psychophysical scaling. Psychophysics is relating psychological perceptions to physical measurements. So we want to know a mathematical scale of the perception as a function of what we can go and measure physically repeatably. And people will use the term subjective. I despise the term subjective because my psychophysical measurements are every bit as objective as the physical measurements. It's just I have to use people to generate them. And I have to do statistics to make them repeatable. And maybe the variance is bigger because people are people, but everybody's got variance. It's just a matter of quantifying it. But I can measure, say, your perception of image quality repeatably over and over again and get the same scale. That's objective. Your personal preference of image quality is subjective, but the statistical response of everybody is pretty objective. So I'm going to show you a quick little experiment. We're not going to do the experiment, we don't have time. Basically, hierarchy of scales. Nominal scale is just naming. My name's Mark. Anybody else in the room named Mark? Good. So if we wanted to measure the height of everybody in the room, I could say my height is Mark. And everybody else has a different height because they have a different name than me. That's a nominal scale, not terribly useful. Because if another person walked in here and said, hey, I'm Mark, and we'd say, oh, you're the same height as that Mark, because we're measuring height by name. Wouldn't hold up very well. Ordinal is a scale where you can put things in order, greater than, less than, but we don't know intervals. We don't know how much greater than and less than. Interval is one of my favorite ones. We know how much greater or less than now, so we can do addition and subtraction, but we don't know where 0 is. That pesky 0 is unknown. So we just have intervals. We don't have ratios. We can't do divisions. Temperature, other than the absolute temperature scale, is measured on an interval scale. Celsius and Fahrenheit have an offset relative to each other. 0 doesn't really mean anything on either of them. So what about the freezing point of water and boiling point of water? That's arbitrary. They're interval scales. Ratio means we know where 0 is. We measure length on a ratio scale. 0 inches means something. 6 inches is half as long as 12 inches. The way we really measure height. A lot of times in perception, we're up here, we want to get down here. That's what the scaling is about. The color matching I talked about is really a nominal scale. It tells us 2 colors match or they don't. If those 3 numbers, the tristimulus values, are equal, colors match. If they're unequal, they don't match. And just from those numbers, they're so nonlinearly related to perception. And the banana, remember the banana? I can't tell you what color it is just from those 3 numbers. I need more information to factor in. If you know French, this is the word noir. If you like wine, Pinot Noir, my favorite. It's a good way to remember these scales in order. That's all I got for math teaching. Quickly how we make interval scales. So we use this thing called the Law of Comparative Judgments that was published by Thurstone in 1927. And basically he said, I do it this way because we usually put our psychological scale on the y-axis. Stimulus I is up there with some distribution, J is down there with some distribution. If we know the distribution and all we ask people to do is say, is I better than J, worse than J, brighter than J, more colorful than J, redder than J, whatever question we want to ask. If we get the proportion of responses above and below, and we know what the distribution is, we can put them on the scale because we know what the probability of them being greater or less than, as long as they're not too far apart. This is also called confusion scaling. If there's no confusion, you now have an ordinal scale. All you know is they're completely different. Is red the same as green? No. To say how close together they are, we have to compare red to a little less, maybe yellowish red, and so on all the way around to get to green. There has to be some confusion to build the scale. So we do this. Here's the equation for the difference. It's based on the z-score, which is, if we're assuming a normal distribution, the inverse of the normal distribution, the variances for each thing and the correlation between them. As you might imagine, being on the physics end of psychophysics, we assume correlation is 0, and we assume the standard deviations are equal, and all that goes away except the z-score. It becomes a constant. That's what we do in most practical cases. The experiment, this is z-scores. There's other versions. The experiment we usually do is called a paired comparison. Here's two things. Which is better? Which is greener? Which is brighter? Easy task for the observers. That's why I can say it's objective. You'll pretty much give me the same answer all the time, depending how close together they are, of course. The downside is if you have 5 stimuli, you have 10 pairs. If you have 20, you have 190 pairs. So if I want to compare 20 things, I have to show you 190 pairs and make you do that judgment. That's where the brownies come in. The brownies are what we give the observers after they do the experiment to get them to come back and do more experiments. The cookie kind of brownies. What kind? Good ones. The better they are, those grad students get more observers. So this is what an experiment looks like. This is one I actually run in a class. And I've done it both on a display and with little prints. It's image quality. I take 2 images of this mountain side by side and I say, which one do you think is a better quality image? And I have 5 levels of contrast. I've varied them in lightness, it turns out. And we go through all the pairs. And just to give you an example, we keep score for each pair. This was experiment 2 in a class, that's what the 2 is. Pairs 1 through 10. You would circle A or B, which one you like better. And show you all the stimuli. These are very blurry because these were made a long time ago and they were printed about this big for the students to take home. But you could look at this and say, well, I like a little more contrast in my image. I like B better than A. And we go through all of them. You can see they're pretty big. It's not a hard task. Pretty big differences. You would say A or B on all of them. We get done. I had 5 different pairs, so this is my answer sheet. 3 and 4 were the first ones, and so on. There's my physical variable. It was a sigmoid applied to L star, a lightness equation, and the values. I analyzed my matrix. So just a point here. 2 was preferred over 3 by 12 people, and 3 was preferred over 2 by 6 people. I had 18 people in that class. 18 people did that task in a little more time than we just flipped through it. I gave them time to answer. Convert those to percentages. Across the diagonal, they're equal, or they add up to 1. So 2 over 3, 67% of the time. 3 over 2, 33% of the time. The other part. So we call a forced choice. You have to pick. You can't say, I don't know. Some people hate that, but they get used to it. And the thing is, if you really don't know, half the people are going to pick one, half the people are going to pick the other. You randomize everything. It comes out 50-50. Some of them are pretty close, even with just 18 people. Convert those to z-scores. So now they're across the diagonal. 2 compared to 3 is 44. 3 compared to 2 is minus 44. The minus is saying the direction. 2 is here. 3 is here. 0.44 units apart. Plus or minus, depending which way you're going. That's the distance on the scale, assuming people were normally distributed. We average those to get our scale values. We plot them. We put error bars on them based on the size of that standard deviation and the number of observers. And we often get a plot like this where somewhere in the middle is the most preferred image. People don't like images that are too flat, and they don't like images that are too contrasty. There's an optimal number. And you can see those error bars are pretty big, but that experiment was really simple. And only 18 people to generate this. When I do this class after class, the curves always look like this. They would plot right on top of each other, not perfectly, but within those error bars, as you'd expect. And then you get enough observer, those error bars start shrinking. And that's useful if you want to make a new display or something and know where to put the contrast. Okay. 5 minutes, and I only have about an hour's worth of stuff left. Never been in that situation, right? So I'm gonna go quick. I knew I had too much, so I'm not gonna try to cover it all, but I'll flip through it. Multidimensional scaling is a similar process in more than one dimension. I love this example, so if you haven't seen it, I will spend time on this example, then I'll quit. You can do the same kind of experiment, compare similarities of different things, and then see how many dimensions it takes to describe them. So the USA example is, remember in the old days when you had road atlases? I used to
Still
love
have one.
reading road atlases. We still do have them. But in the back there's usually a matrix that says, I want to go from Rochester to Albany, it's 300 miles or whatever it is. That's paired-wise data on the distances between places. So you take that, I took a bunch of cities, Atlanta, Boston, Chicago, Dallas, Denver, LA, Seattle, New York City. I'll note right now, I was sitting in Rochester when I picked these cities. And in my mind, I thought, I'm just gonna kind of equally space them out in the US. Now, I've driven across the US several times. I know how far it is from Seattle to LA, but that didn't bother me sitting in Rochester. They're right next to each other. So think about that. But here's all the miles. That's all I put into a multi-dimensional scaling routine, is this matrix. And I say, what space does this fit in where all of this can be true? Is it a 3-dimensional space? Am I flying through outer space to get between these cities? Is it 2-dimensional? The USA flat? Is it 1-dimensional? Are we all on a line? So you put it in. You make what's a similarity matrix typically. So higher numbers are closer together. It's just the inverse of the miles. There's different ways to do it, assuming linearity or not. You have an objective function, which is the stress. It's the difference between the fit and the data. Just like any fitting function, you minimize that with the optimization. This shows the minimization, the stress in each number of dimensions. In USA, there's some stress in 1 dimension, there's almost no stress in 2, and it doesn't get any better. Typical 3D configuration, like color space, lightness, chroma, hue, it'll come down pretty close to 0 at 3 and stay there. So where this function levels off is where you say that's the number of dimensions in the data, and then you get the configuration. This is the fit, the goodness of fit. So in 1 dimension, the US cities don't fit so well. Not bad, actually, but in 2 dimensions, they fit pretty much perfectly, as you might imagine. And then there's different ways to measure distance. We do this all the time. In color, distance isn't always Euclidean. Sometimes it's what we call city block, where you say, I'm going to look at the lightness difference, then the hue difference, then the saturation or chroma difference. We use different distance metrics. Here's the US in 1 dimension, Boston, Seattle. Whole country's in a straight line, right? That's how it works. Doesn't look quite right, but at least we got east-west. We got the main variance in the first dimension. If you've ever done or heard of principal components analysis, this is very similar. The first dimension of variance is the first dimension of the MDS. Here it is in 2 dimensions. The algorithm doesn't know anything about east-west, north-south. It just knows distances. So it's going to spit out whatever dimensions explain the variance the quickest. So it doesn't quite look like our map, but it kind of does, except it's flipped over and flipped top to bottom, left to right, east, west, north, south. So I can flip it over, no problem. These are just dimensions, they're unitless. Well, I can put units on them. And there we have it. Boston, New York, Chicago, Atlanta, Denver, Dallas, Seattle, Los Angeles. And you can see as we got farther away from Mark and Rochester, the cities got further spaced, which is another perceptual thing. When you have more of something, distance, it takes a bigger change to notice a difference. So when you're sitting in Rochester, LA and Seattle aren't that far apart, even though they really are. If you're sitting in Seattle, you know it. The other thing is the way the country was settled, the cities are farther apart out west. So there's 2 variables there. And we can fit the dimensions. And this one was easy because the first dimension is east-west, second one north-south. I can do a regression and see how good they fit. I don't think I included that. I didn't. But we can figure out what we're looking at. In color, this would be our perceptions. So this talks about scaling lightness. We do it in all 3 dimensions to build these color spaces where I can start to say the banana is green, not just what matches the banana. And that's what all the rest of the slides are. I'm going to go through them quickly because I might have a conclusion slide. Big model takes more variables like the color of the transparency I put up into account. Lots more math to do it, lots of empirical stuff. But I'm gonna stop with that. I think you got a sense of what we do. I'll bring the lights back up just a bit. Well, sorry, I usually try to do that slower with a warning. But I just wanna remind you this last quote that came from Munsell. We had a conference on Munsell last year in Boston. So I did a talk that was basically Munsell's impact on the color world, and found this quote, which I like: "Color's in us, not outside." So it's a perception, it's not just the physics. Thank you, I'm out of time, but you have